What's up YouTube, Professor McCullough here, and as you can see, I'm in my brand new, nice, big, beautiful office. And this office has a nice big window where it has impeccable lighting. So a lot of my videos are gonna start coming from the office now, if that's okay with you guys. Now that being said, you guys clicked on this video so that you can learn about the 4 macromolecules: proteins, carbohydrates, lipids, and nucleic acids, right? Well, without any further ado, let's go ahead and get started. Now, virtual class. The first thing that we have on the list are proteins, and you can pretty much off the bat tell if a molecule is a protein based on the ending of the name. If the name ends with IN, then more than likely it's a protein. So we have immunoglobulin, hemoglobin, keratin, and those are just some general examples. Now, proteins have a structure that you can see in this picture. On the left, we have what we call an amine group, which is usually depicted as a nitrogen and 2 hydrogens. On the top, we have the hydrogen group. On the right, we have the carboxyl group, which is a double bonded carbon to an oxygen and also another oxygen. And on the very bottom, we have what we call the side chain. And the side chain is what's going to tell you the amino acid. Now, there are hundreds of amino acids, but in the human body, we only use 20, okay? And I'll probably do another video talking about the amino acids, so somebody remind me in the comments to do that. All right, and the difference between the amino acids, like I said, is really the side chain. And these actually, they combine through a process that we call dehydration synthesis. All right, and when they combine, they form what we call a peptide bond. So a protein is essentially a bunch of these amino acids, and they're combined with a peptide bond. And when you have 2, that's a dipeptide. When you have more than 2, then it's a polypeptide. But the proteins we think of usually are hundreds of amino acid strings put together and folded in different shapes, okay? Speaking of protein, I was about to say protein shakes. Speaking of protein shapes, you can kind of, when you're looking at a protein, you can tell based on how much you're looking at and what kind of protein shape it is, basically what level the protein is. Like, for example, if you're looking at a protein and it just shows the amino acid sequence, meaning the carbons and the nitrogens and the side chains, you can actually literally see what atoms are in there. It's called primary structure. Now, when you have a large quantity of those, then they start to do things such as form into a spiral, or they could actually form into sheets. And we call the spiral, we call that alpha helixes, and then we call the sheets, we call them beta sheets. So if you're looking at a picture, and your teacher says, okay, this is a protein. You're looking at the picture and then you see the alpha helix or the beta sheet, then you can say, okay, cool. This is what we call secondary structure. And then we have tertiary structure, tertiary meaning third or the third level. And tertiary structure is usually when you have multiple alpha helixes and multiple beta sheets, and they kind of squished up into one. And last but not least, you have what we call quaternary structure. And quaternary structure is basically the entire protein with all the alpha helixes and beta sheets. But see, sometimes some proteins have so much of the amount of amino acids that you have to actually depict the different actual chains of the amino acids. And a lot of times they're held together by things such as iron, for an example. And when you see the things that hold it together and also multiple chains, multiple alpha helixes and beta sheets, we call that quaternary structure, okay? Now moving on to carbohydrates, okay? Now carbohydrates, in other words, you could say they're basically sugars, right? And they're also could be called saccharides. Now, if you ever seen a commercial, I remember when I was little, it was a commercial and it was basically saying that maltose and fructose and glucose are all the same thing. Well, no, that is false. Okay. Now, first of all, let's look at the ending of those words. So you can pretty much right off the bat tell something's a carbohydrate based on what we say O. So OSE. All right. Glucose, cellulose. All right. So most of the carbohydrates, they usually end with OSE. Now, carbohydrates are very, very, very important as far as energy, especially in living things, because that is what we're going to break down to convert into our cellular energy. So when you see a molecule and it has twice of the amount of hydrogens than oxygens and usually carbons, then we can pretty much assume that that's a carbohydrate. For example, glucose has 12 hydrogens, okay, and it has 6 oxygens and also 6 carbons. See, carbohydrates could also form in strings, or they can form in nice ring hexagon shapes or pentagon shapes. See, these are also linked by what we call dehydration synthesis, all right? And you can have one ring, or you can have, for example, in plants, the cellulose, you can have hundreds and hundreds of rings, and they're usually classified by how they branch out. If they branch out linearly, or if they branch out more like a tree type of shape. Whew, you guys doing good so far? Not too bad, right? Okay. Next we have lipids. And lipids are very interesting because the best example I can give you of a lipid is like your earwax. All right. By the way, don't stick Q-tips in your ear. You can damage your ear canal. It clearly says on the box. You can pause the video and go and check. But anyway, earwax is a good example of a lipid. And a surefire way you can tell that something is a lipid in your body is that it doesn't dissolve in water. Okay, so imagine if your earwax did dissolve in water. You would go swimming, right? And then your earwax will dissolve in the water, will get all up in your eyes like this, okay? And then basically drown you from the inside out. And that probably wouldn't be pretty. Also, things like beeswax is a good example of a lipid that you can find in nature, but also the phospholipid bilayer, because it keeps the water separated from the intracellular space in the cellular membrane because it can't be dissolved, okay? And you can usually tell a lipid is simply, the formula is simply what we call a glycerol plus a fatty acid. The fatty acids are important because they're going to tell us what type of lipid it is as far as what it does at room temperature. Meaning most plant oils are the equivalent of animal fats, okay? So these fats are stored and they're usually saturated, meaning that they're nice and linear and they can stack on top of each other. Versus plant oils, which are usually unsaturated, which meaning that they're more fluid, okay? And they're unsaturated because of the presence of a double bond in the hydrogen. Last but not least, you have the granddaddy of all molecules, right? You have the nucleic acids, all right? And the nucleic acids are simply RNA and DNA. And now if we look at the name DNA, deoxyribonucleic acid, okay? So where the ribo is coming from the ribose sugar, which is a carbohydrate, which I mentioned earlier in the video. Also, you have the deoxy, meaning that ribose sugar is missing an oxygen, it's deoxified. So deoxyribo, and then nucleic acid because they're nucleic acids, all right? So DNA is responsible for telling the organism what to grow up into. So everything that you see is the physical manifestation of that little double helix molecule in your cells, right? That DNA, all right? Now, if you have to test something, just remember that these DNA sequences, you have 4 different types of what we call base pairs. So when you have a base pair, A always goes with T, C always goes with G, okay? And then that ribose that I was talking about, and also a phosphate, right? So that ribose sugar and the phosphate, they form like a backbone on each side of the base pair. And then the result is like a ladder formation, okay? And the DNA helix is kind of twisted, right? So it's like a twisted ladder or a staircase type of thing going on, okay? And also you have the RNA, and RNA is kind of like half the DNA, all right? But see, instead of T, it has what we call U, okay, uracil. And the RNA is mostly responsible for being fed through the ribosomes, and it's going to be a video about the organelles coming soon too. So the ribosomes take the RNA and they read the codes on that. So every 3 base pair, that's going to code for a certain amino acid and it's going to spit out the protein. Well, I hope you guys enjoyed that quick review on the 4 macromolecules so that you can impress your professors when you get back in class. And I'll catch you soon for another video.